So our last speaker in this section of the conference today is Gordana Vunjak-Novakovic. She's the Mikati Foundation Professor of Biomedical Engineering and a Professor of Medical Sciences at Columbia University. She obtained her PhD in Chemical Engineering from the University of Belgrade and was a Fulbright Fellow at MIT. The focus of her research is on engineering human tissues for application in regenerative medicine, stem cell research, and modeling of disease. She's extensively published and highly cited. She has over 70 licensed, issued, and pending patents. She's been the co-founder of 2 biotechnology companies and is a frequent advisor to government and industry. She's an elected member of the American Institute for Medical and Biological Engineering, as well as several European academies of science and engineering. And she is the first female professor at Columbia University ever to become a member of both the National Academy of Engineering and the Institute of Medicine. Please join me in welcoming Gordana.
Okay, so this is really a terrific meeting, and I'm pleased to be here with you. I'll talk today about tissue engineering. This is really something I'm interested in. And what I would like to tell you about today is that I think we are, in fact, witnessing the 3rd generation of this very exciting field. I'll tell you a little bit about motivation and logic, and then I would like to use one example, one single story, to illustrate some of the capabilities that we have developed as a field over the last 25 years or so. So the first question that everyone is asking you is, why is tissue engineering important? And why is it attracting today some of the greatest young talent? I would like to offer you 2 reasons. I mean, one is that we live longer and we live better than ever before in the history of humankind. So we need more maintenance. We are members of an aging population, and you may compare old people in a way with old cars. You know, things break every now and then, and you need to fix them. And this is exactly where engineering for medicine, tissue engineering, can help. And the other reason is that tissue engineering can foster medical research in many ways that go way beyond regenerative medicine. A couple interesting facts. About 2,500 years ago, during the golden era in Greece, the life expectancy for humans was only about 28 years. And over more than 2,000 years, to the beginning, maybe the end of the 19th century, it only increased from 28 years to about 40. And this is because medicine was developing, it was taking better care of people in many ways. And then over 100 years, it doubled. It's today 78 or 80 years in the United States. You can take another 2 perspectives at the same numbers. One is that among all people who ever made it to age 65, 2/3 are alive today on Earth. So this is really dramatic. In 1920, 1930, also in this country, you would have about 40 people working per 1 person retired. Today is almost 2 to 1. So this is just like a milieu we live in. And this is one huge motivation for looking for options for enabling people to be more functional and to live not only longer, but also better lives. So failure of an organ is one of the most devastating conditions that one person can experience. And if your heart or your bone or your lung or kidney or any other organ fails, then there are a couple different options one can pursue. You can obviously reach out for a mechanical device of some kind, which can only provide part of the necessary function. And in an ideal world, we would really like to have the missing or damaged organ to be reconstructed, to be replaced by a biological substitute that would provide the entire structure and function. So looking a little bit back, in late 1980s, there was, in one of these Christmas issues of a magazine, it was a Life magazine, there was a story called Replaceable You. So they interviewed scientists, clinicians, to see what do we have available at that time to replace damaged or missing tissues. And you can see this Robocop here. Obviously, you cannot see detail, but absolutely everything is artificial. You see wires, you see ceramics, you see plastics. So this is what we had available at that time. So this was only 25 years ago. And then coincidentally, 10 years later, another magazine, Time magazine, was running a similar story, and the outcome is strikingly different. You see that here, everything is biological. So this doesn't mean that in 1999, we had capability to engineer and replace all these tissues and organs. It really means a total change in the mindset. People were looking towards a book of biology, getting guidance to design systems that would be real biological replicates to the maximum possible extent of our lost tissues. And today we come, I believe, to tissue engineering 3.0. And there are two important factors that actually are leading into this third phase of tissue engineering. One is the advent of stem cells. This little cartoon really tells a message. I mean, the parental advice, parent stem cells are telling to their child stem cell, you can be anything you want when you grow up. And this is exactly true. I mean, we have technologies today to isolate adult stem cells from a person from, say, droplet of blood or a little lipoaspirate, a little small amount of fat, and then drive these cells, direct them to differentiate into many, many different lineages. And the other driver is technology. And I'll show you some specific examples about it. So these two reasons are actually development in stem cells, development in technology, have actually transformed tissue engineering from something that one of my students called like a tissue what. She said, like, this is really not tissue engineering. This is like tissue, try this. So there is much more like a more logic, more directed, rationalized approach than we had 20 or 15 years ago. So the logic of tissue engineering is remarkably simple. So cells are the tissue engineers. It's none of us. I mean, the only entity that can build a tissue is a cell. So what we do is not really to engineer tissues. We engineer or design the environments that are instructive to the cells so that we can tell them what exactly we want them to do. So this is how tissue engineering really works. One other important component in this whole process is actually technology. So when you look into how we approach tissue engineering today, this is presented in this little cartoon. We approach the process in a way that is personalized, that is really tailored to the patient and to defect that you're trying to treat. So the patient is source of actually two streams of materials and information. We would always start from the patient's own cells, because this is giving you an opportunity to actually maintain the genotype to avoid all and any problems about disease transmission, incompatibility, and whatnot. So we would take the cells, the tissue engineers from the patient, and then we would design two components that are essentially directing the cells what to do. One component is the biomaterial scaffold. So this is a template. This is a structural and informational template for tissue to form. And the other component is what we call bioreactors. So this would be a culture system, which is the source of nutrition and regulatory signals. In order to make something in an anatomical shape that we need for eventual replacement in human, we would use imaging data, clinical imaging data, to fabricate the scaffold with its external shape and internal structure, and also to fabricate an appropriate chamber of the bioreactor system. I'll show you an exact example on how this is done and used together. And if the conditions are right, if we transmit the appropriate messages to the cells, we would get functional tissues such as we work on bone, on heart, on lung. I will show you just one example today. It will be about bone. The same approach, the same fundamental approach of assisting the stem cells using scaffolds and bioreactors to become functional human tissues is also being used in organs on chips. So we make very large numbers of very small tissues on these platforms that are specifically designed to enable drug testing. And these are usually multi-organ platforms because you're really looking into interactions, say, between vasculature and heart and liver when you're looking for cardiotoxicity. And we also use the same engineered tissue, just in smaller size, to study disease. For example, we are building models of tumor metastases in bone and lung. So there are many, many opportunities how you can use technology to advance biological research. So this was what I meant when I said that tissue engineering can drive progress in medicine beyond regenerative medicine. So let me just show you one example of a technology. I will talk about bone. So bone repair can be a huge problem in medicine. If you have a small defect in otherwise healthy bone, it will heal. This is one of the tissues that heals beautifully and completely. But if the defect is large, if it's beyond certain critical size, and we are talking about millimeters here, not about something much larger, or if the bone is totally missing due to either congenital defect, which you see, this is the situation here, you know, some bones are just non-existent, or you have a patient, I mean, you know this face here, who underwent multiple cancer surgeries and stayed without some crucially important bones in their head, then nothing really helps. And in an ideal world, you would really like to regrow this bone. So we started to look some 10 years ago into the head and space region, because this is where you really challenge your engineering skills the most, because face is what makes each of us different from another person. And this is where it really matters with what kind of fidelity you will be able to reconstruct the bones and cartilages and muscles and whatever else is damaged. And this is also, like any damage is really bad, but this is also bringing not only physical problems and physiological problems, but also psychological problems. So we sort of decided to tackle one of the challenging areas. And the approach that we proposed is really a variation of what I just showed you. So we started to work with one particular graft. So this is this whole upper part of the lower jaw, because this is anatomically extremely complex. You can see here our scaffolding material. It's different in each dimension. And it is also the most loaded bone in the head. So we reasoned if we can make this, then we can also make the cheekbone and the skull bone and maybe some other bones of the head. So as I told you, imaging was the source of data that we used to fabricate biological scaffold. And we also used this data to fabricate the chamber of the bioreactor that was exactly matching the scaffold. So it was embracing it so that we can move the fluid through the scaffold, which is seeded with cells. And the cells also come from the patient. So you actually load the scaffold into the bioreactor, infuse the cells, let the process of conditioning go for a while, and then you get a bone graft that you can study further and eventually implant into a person. So talking about materials, because this is really in the title of this session, we have like a little enterprise running in the lab as a separate direction, which is to make biologic scaffolds out of human tissues. So far we derive materials from 14 different organs that are schematically shown here. Whole organs, specific regions, healthy or diseased. For example, you can have a relatively healthy region of the heart, or you can have the exact area of the heart infarction. So you can study the responses of matrix of different properties. We also get this from animal tissues, transgenic animals, and whatnot. There are 4 different formats, which actually gives you a lot of options for research. We have intact tissues, so the cells are removed, but the matrix is completely preserved, so most of the composition, architecture, and mechanical properties. We can turn this preparation into solution, so you can add this solution into your culture medium if you only want molecular effects. We can turn it into hydrogel, and we can use it to reconstitute something 3-dimensional in form of a sponge or something. So for bone, we really use the preparation from the bone itself. So our scaffold was the bone itself. The bioreactor that I mentioned is shown here. Just to get an idea, you see the bone scaffold inside this white thing. And a lot of engineering went into designing this bioreactor to understand these are the flow paths, how exactly to perfuse these structures so that all cells get the right amount of nutrition, oxygen, and shear. Shear is very, very important for the bone. And here is a little movie that just shows you where we go. So we take stem cells from the patient, we take images to fabricate scaffold, put scaffold into the bioreactor, infuse with cells, grow a piece of bone, and then we envision that this will be implanted into the patient at the end. So at some point, maybe 3 years ago, we were at the point that we thought that we have a technology that may be applicable to human condition, but our NIH funding is really for research, not for translation. And you all heard of Valley of Death. And we got one of these phone calls that was totally transformative to us. I mean, in the introduction, this was mentioned. So there is the New York City Partnership Fund that has a so-called BioAccelerate program to bridge from the point where you are like all done from the NIH perspective, but still too early for investors, to the point when you may be at in the right place. So they actually gave us funding for the large animal study to study technology in a human-scaled model. And actually, this led over these last few years into us starting a company, raising funding, getting another infusion from the investment fund in form of cost match. And actually, we are now like full power in preclinical studies of this technology. So the approach, what we have done with this money is everyone, of course, volunteered our time. We just used all money for the animal study, and we calculated how many pigs we can implant. Pig is a perfect model for craniofacial work in human. So each individual pig was imaged to make a scaffold in bioreactor chamber and donated a little bit of fat. So we work with fat tissue because this is the tissue that you can sample most easily in humans. And then after the grafts were cultivated, so this took 3 weeks of conditioning, we implanted these grafts into the pigs, left them for 6 months, and evaluated over this time using imaging at multiple time points and endpoint assays at 6 weeks, 3 months, 6 months. So because, as you know, everything is unbelievably expensive in this city, so to maximize what we can do with a given amount of money, we actually worked with our collaborators in Louisiana. So the pigs were in Louisiana. Images and stem cells were traveling to our lab. We were making grafts, and then the grafts and ourselves were traveling to Louisiana for implantations. we have to do an additional engineering work for this study. So 2 things that we did to modify the bioreactor that I showed you. First, we made it geometrically different so that it can fit into imaging device. So what you see here, this is a photo of the actual bioreactor. It could fit into micro CT, and then you can see how you're doing without interrupting culture in any way and take it out. So this helped us to really optimize the time of conditioning. And also, this kind of bioreactor was modified to work with a battery-operated pump to be constantly perfused. So we could package a number of them into a sort of attaché case contraption that we brought to Louisiana. So we were testing, actually, the feasibility of our idea that the grafts will be grown centrally in some place in the United States and then transported to hospitals around the country. So I'll give you just 3 data slides to show you, to give you a flavor of what we have done. So what is shaded in blue is actually the piece of bone, including this cartilage layer that we replaced. What you see here, diagonally, like top right and bottom left, are 2 pairs of bones. The red one is the bone taken out from animal at the time of surgery, and the white one is the one that we engineered in advance using images. So you can see 2 things. One is that actually this engineered bone is similar to the actual bone. And we can go down to some 30 to 50 micrometers in precision using technology we have. You also see that this pair of bones from one pig is very different from the pair of bones in the other pig. And this is the same bone. And the same is with humans. So you do need to customize. You cannot make like a small set of grafts that would fit everyone. And this is just a scene from surgery where we work together with our colleagues from Louisiana. So what I show you here is actually the result of implantation after 3 months, the top panels, and 6 months of implantation. And at the left, you see the control group where we implanted only the biomaterial scaffold. And at the right, you see the group where we implanted engineered bone. So after 3 months, you see that actually this group is more porous. There is no real integration between the graft and the host bone. What you see here is a CT, which means you see only mineral. This is not empty. I'll show you on the next slide. This apparently empty area is actually fibrous tissue. And the group with engineered bone looks better. You see that it is still porous, but it is integrating very nicely. After 6 months, this graft is practically gone. So the graft is being resorbed constantly, but actually the ability to build bone without cells that are infused into this graft is obviously very small. In contrast, the other one with the implanted bone, it looks really strong and very well integrated to the host bone. And when you take other looks at this bone, this is just a sample. So the top left is the mineral. You see very nice structure of the bone, of trabecular bone. The top right is an example of one of the bone proteins, osteopontin. The light areas are the scaffold. The darker areas are the actual bone matrix. Bottom left is the bone histology. This is how you really want it to look. You see that there are marrow spaces, there is vasculature, there is really very, very nice building of the bone matrix. And the bottom right is actually the vasculature. I didn't talk about it, but we do have methods to facilitate pre-vascularization of these grafts, which is very, very important for bone to stay alive. And I would just like in closing to make a couple comments. The first comment, I would like to go to some of the introductory remarks. I mean, I couldn't agree more that this is the most exciting time to work in bioengineering, because we are indeed entering the era of personalized medicine in regenerative engineering. So where we are going is this, the main goal is to grow biological replacements of damaged tissues, essentially any tissue in the body. Today, you have bladder and skin and cartilage and trachea already in clinics. You have heart and lung and bone coming along. There are many, many challenges we are facing, but actually the progress being made in the field is really gratifying. The whole concept of tissue engineering is really to instruct the patient's own stem cells to rebuild the original tissues. So engineering is using biological principles when designing tissue engineering systems. And if you just want to say the end result in very simple words, we are going to some kind of one-stop body shop that is tailored to our individual needs and that will supposedly help us live longer and better lives. Thanks very much for having me.